Rajgopal, Pavin?
Me?
Sorry. Yes. I can. I'm sorry. Yeah, go. Yes, Pavin, go ahead.
Can you see it?
Yes, go ahead. I can see it. It's perfect.
Okay, so my name is Pavan Rajagopal. I live in Maryland and I go to Centennial High School. As a hobby, I like to play tennis, and I play the piano, and I just do science Olympiad. But yeah. So my research topic is exploring epigenetic signatures in Huntington's disease and unraveling the role of epigenetic modifications in disease progression. I just wanted to start by introducing what Huntington's disease is. Huntington's disease, otherwise known as HD, is a neurodegenerative disorder caused by genetic mutation, specifically the expansion of CAG repeats in the Huntington gene. This mutation results in the production of a toxic mutant Huntington protein, which leads to progressive damage in certain regions of the brain. Individuals with HD experience a range of symptoms, including involuntary movements, cognitive decline, and psychiatric disturbances. Today, my research has explored the epigenetic dimension of HD, and my question was, what are epigenetic changes in HD, and how do they correlate with clinical manifestations? This research is crucial for developing the targeted therapies and refining our understanding of HD's complex pathophysiology. So my literature review, I delved mostly into global and local studies, and I had a specific focus on Maryland. I synthesized information on HD's epigenetics, and various theories were examined, which highlighted the complexity of epigenetic modifications in neurodegenerative disorders. I identified weaknesses, which include the lack of standardized biomarkers and variations in research methodologies. I used clinicaltrials.gov, and I also used a couple articles that were provided by Professor Varkey. Moving on to our methodology and results. Through literature review methodology, I meticulously analyzed the existing studies on HD and the epigenetic landscape. I used data collection, data extraction, and data analysis, and lots of detailed information were selected from the various articles that I used, which include study design, participant characteristics, and methodologies for epigenetic profiling. The results were as follows. DNA methylation patterns. The analysis unveiled significant differences in DNA methylation patterns between individuals with HD and healthy controls. Notably, hypermethylation in regions associated with key neuronal functions were identified, which suggested a potential link between aberrant DNA methylation and neuronal dysfunction in HD. There was also histone modifications, and that identified specific marks correlated with dysregulated gene expression. Reduced histone H3 acetylation in the striatum of HD patients suggested a potential role in the progressive degeneration of this crucial brain region. And finally, correlation with clinical manifestations. It offered a nice suggestion association between specific epigenetic changes and clinical manifestations of HD. Individuals with higher levels of DNA hypermethylation in specific gene regions exhibited more severe motor symptoms and cognitive decline. Distinct histone modifications correlated with the onset and severity of psychiatric symptoms. And our conclusion, my research aimed to identify the epigenetic signatures in HD. The findings revealed a correlation between the epigenetic changes and clinical manifestations, which contributes to a nuanced comprehension of HD. While celebrating these contributions, we acknowledge limitations. The primary limitation of this literature review was within the inherent heterogeneity of included studies. Variations in study designs, participant characteristics, and methodologies for epigenetic profiling make direct comparisons challenging. The diversity in sample sizes and demographic factors may introduce compounding variables, impacting the generalizability of the findings across studies. And finally, my research experience with GiftedGabber. Professor Barkey's mentoring helped me significantly on this research topic. He suggested things that greatly changed the trajectory of where my research project's aim was. And without him, I don't think I would have been able to produce this level of research. Professor Virgil's expertise also helped me equally, as he gave really good suggestions on how to format it and the syntax of everything in my research paper. This overall research program helped me overall, as it has given me an introductory dive into the research process and everything that makes it up. I'm truly grateful for this inside knowledge. And finally, the most thrilling aspect of the research was reading through the sources, as there's lots of really cool information that I realized that I wouldn't have realized without reading through all of these. Thank you.
Yeah, you want to stop sharing?
Yeah.
Okay. Dr. Jobin, questions for Pavan?
Pavan, it's a good presentation and it's very close to what I do currently. Huntington's disease, like quite a bit of focus of my research, my current research focus. And for everyone who is listening, role of epigenetics is being increasingly recognized, and then not only in Huntington but other proteins as well. So you might also want to check out that as well for your further, investigation or out of just like curiosity. And Pavin, question for you. So, when you looked into clinical.gov and all, so how do you think that they are developing, or what kind of therapy they are developing against, these kind of epigenetic changes? What are the scientists proposing?
First is like what the basic research they need to understand, like the role of the Huntington gene and the mutation causing HD.
Uh-huh.
Okay.
So I mean,
yeah.
Yeah, please go ahead.
What were you about to say?
No, have they also looked into some kind of therapy where like they look into enzymes which actually add a methyl group to DNA or to histone?
I'm pretty sure they did. Based the article that you sent me, that one thing that it did.
Okay. Okay. Good job.
Yeah. So great. I just want to remind everybody that this is the experience. Working with a professor and expert like Dr. Jobin just elevates your learning. It just pushes it to the stratosphere. And that is what is the opportunity here. You're doing something out of the four walls of the classroom. And I really want to congratulate each and every one of you. I don't think you signed up for the program. Your parents signed you up and you had to take it on, but still you did a phenomenal job of doing the research, showing up. I know it must have been a struggle. You must have felt like Alice in Wonderland, but that's what brings in change and invention, right? Status quo brings nothing, right? So I want to give a big round of applause for each and every one of you. You are phenomenal. Okay.